The question was, would you find that most people would weaken to malonaldehyde? No, but a lot of people do. What would make me go along this pathway would be anybody who's strengthened to vitamin E. So a person who strengthens to vitamin E, so what you may want to do is to upgrade some of your kits. If you've got kits and you've got a simulator, what I would do, I've upgraded my vitamin E simply by using the same vial and putting wheat germ no-sode in it, the marker for wheat germ, and zap it in. Then you sort of upgrade it, okay? Rather, like I said, upgrade your homocysteine to load it with methionine, you'll pick out more people, okay? So you can upgrade if you like. So on these cases, we'll teach you which ones to upgrade. And certainly like vitamin D, in the old kits that we used to do, vitamin D never showed up with patients. I think I had one in 5 years or something that showed to vitamin D. But when you upgraded it with the hemp seed oil, you know, half the nation over the winter showed to vitamin D. And it's the same you'll find with the CoQ10. I really can't remember more than half a dozen people I've prescribed CoQ10 in the last few years to, really as far as basic CoQ10. But when you upgrade it with the right oil that it carries it across the gut wall with, you know, I've always read that the oil-based CoQ10 was much better. It's amazing. You know, then you find 10-20% of particularly the heart patients show to CoQ10.
With the wheat germ oil, you take that in the morning?
Muscle test which one? The answer usually is yes. Yeah. It's not like an oil. You're not using the oil as a, you know, an oil source for phospholipids. You're using it as an antioxidant. I just put it onto bread. So when you've made your nice new loaf that you'll be setting to kneading on Monday, all right, okay, and you put your nice oils in there, then you could, what I just do is I cut myself a slice in the morning and pour the oil on it. Pour the vitamin E. That's the way to, you know, have it. You can put it on a spoon, swallow it down, and so on. But, what? Nice. Yeah. There's no. It's not a particularly pungent oil, but they've had. Yeah. Yeah. It's very bland. See, it's not bad on a spoon, but it just spreads onto bread very nicely. I wouldn't put it onto hot toast quite so much because you might oxidize it a bit. Put it into
smoothies.
Yeah, of course. Yeah. No. Would a wheat allergy, a true wheat allergy, affect it? No, because you're looking at oil and not protein or lectin. You know, you'd be fine. I'd lay my reputation that you would be fine on wheat germ oil, okay? But if you've got a person who's, say, a true celiac, they may be reluctant to do it. But I think they'd be fine. It's like people with peanut allergies seem to be absolutely fine on peanut oil. But, you know, trying that out when they've been really seriously ill with anaphylaxis is a bit dubious, because sometimes you get protein residues, and people with peanut sensitivities tend to be very sensitive, don't they? Much more than a gluten. But muscle test it. Yeah. None, I don't think. True ones. You get a lot of people who've got diarrhea to eating wheat, but you know, with the rancid fats that they're taking in the wheat bread and things, and like we saw with the shortbread, it's not surprising the body wants to get rid of it. You never had this. See, this was the issue that I discussed with this guy from the grain store, because he said it's the complications of the modern wheat with the gluten and the different proteins that you're building up the antibodies to. If you had the natural wheat, they probably wouldn't. You never heard of people having problems with wheat 50 years ago.
And people find that they can eat wheat if they go to France.
Yeah, that's right. Or Italy, they can eat French bread,
Yeah.
you know, which is really, there's nothing to it, is there? And that's why they don't have the reaction, because there's nothing in there. One mouthful and it's gone, isn't it?
And they eat their bread every day. They cook bread every day.
3 times a day in France, yeah, 3 times a day. Okay, so that's just a little bit about rancid fats. So reactive oxygen species degrade polyunsaturated lipids, in other words, free radicals, forming malondialdehyde. This compound is a reactive aldehyde and is one of the many reactive electrophile species that cause toxic stress in the cells and form covalent protein adducts, referred to as advanced lipid oxidation. What that basically means is if somebody, in my experience, shows positive to wheat germ oil, vitamin E, then they've probably got lipid peroxidation. Okay? So in other words, they're going rancid. Okay? So what they need is to increase the amount of vitamin E, and that may be the quickest way and the best way, is the most natural way, is wheat germ oil. Okay? Because all other oils are way down there. Okay? And remember, soy derivatives, which is where commercially, you'll see a bit later on, soy contains certain tocopherols, but not all of them. Okay? The great thing about wheat germ is it contains the 4 tocopherols and the 4 tocotrienols, which are more active than the tocopherols, whereas some plant oils will only contain certain ones and not the other ones. So wheat germ is a very, very good one for many, many reasons. So I'm going to... Trans fatty acids we've looked at a lot in the past, and hydrogenated vegetable oils, the largest scale application of hydrogenation is the processing of vegetable oils, which means basically they're hardening them like we saw with the margarine. And what was fascinating to me, the degree of hydrogenation or hydrogenated is controlled by restricting the amount of hydrogen reaction temperature and time and the catalyst, nickel. So very important for green people to eat butter and their oils, you know, but not bad oils and not vegetable margarines. And this was the thing that came out last month, I think it was. Link between omega-3 fatty acids and increased prostate cancer risk confirmed. Omega-3 supplements taken by millions linked to aggressive prostate cancer. Men who take omega-3 supplements at 71% higher risk of prostate cancer. Omega-3 supplements may trigger prostate cancer. Hold the salmon. Omega-3 fatty acids linked to higher risk of cancer. That was all the stuff that came out. Usually when you find, boom, there's a big press all over the place, it's a press release. It's not usually news. Okay, there's a release. Every day in a certain newspaper in this country, a tabloid newspaper, there's new drug for arthritis, new drug for diabetes, new drug, in its headlines, okay? And it's only in that newspaper. Nobody else carries it. And you know that that's a press release, okay? It's by a pharmaceutical company. They're releasing it. Next day, nobody mentions about it. But every day you think, oh, there's a cure for arthritis. And then you really read it, and it says, sort of states, there's one last week, cure for back pain. One jab or something was going to cure people with back pain. And then you read it, and it's certain types, and it was a salt injection, wasn't it? Then they admitted it doesn't actually matter what you inject into it. It actually opens up the nerve root. It could be just plain water in there. So, and it may help some people, but you blow it up in that it's going to help all those people with back pain. The dreadful thing if it was, because all us osteopaths and chiropractors would be out of business. So a recent case cohort study found that men with higher blood concentrations of omega-3 fatty acids had a 44% increased risk of developing low-grade prostate cancer compared to those with the lowest levels. Specifically, higher blood levels of omega-3 DHA correlated to the higher prostate cancer risk, while no correlation was found for EPA and alpha-linoleic acid. They also had a 71% higher risk of developing the high-grade prostate cancer. The elevated blood levels of DHA found in the feature study is not necessarily indicative of a higher fish consumption. In fact, low-fat diets can also increase DHA levels in much the same way omega-3 supplementation can. While the researchers warn that fish oil supplements may be dangerous, based on the fact this study cannot show causation. Furthermore, no fish oil supplements were actually given as part of this study. That's interesting, isn't it? So they've killed the fish industry, the fish oil industry, without actually giving any fish supplements to these people. So all they found is a high level of DHA. Okay? They haven't, and they've associated DHA with fish oil, not with plant oils and things. Furthermore, no fish oil supplements. Foods rich in omega-3 fats have previously been shown to prevent prostate cancer from spreading. And one recent meta-analysis found that fish consumption was associated with a 63% reduction in prostate cancer. Interesting research. Depends who you're working for, really, isn't it? That's usually a sign when they try and kill off a natural substance, a food compound thing, there's about to be a launch of a new medication. So what I want to do for the rest of today, probably, is to review, or certainly to start, and review the cancer part what we did. Probably the biggest creator of ill health and mortality in this country would appear to be cardiovascular disease, the heart. Now, interesting, again, there's a bit of conflict here because newer research has said it's actually lung disease is the biggest in the United Kingdom. It's overtaken heart disease, and it's swiftly followed by cancer, which is swiftly followed by stroke. So if we add cardiovascular and strokes, because there's great similarities together, then that's probably top of the charts. But cancer is certainly there, and it's very interesting how cancer has increased dramatically in the last few decades as far as the incidence. You know, since the war, or since the beginning of the century, when it was really only 1 or 2 people in 100, and then it went to 15, and now it's up to 30 to 40 in 100. And that was the most common cancers in 2010, because we don't have really up-to-date information, but it probably hasn't changed drastically. And the purple one is females, and the male is blue on here. It's so small, this, I decided not to put it in your manual. But the top one there, as you can see, is breast cancer. Okay, breast cancer outstrips anything else. Second one is prostate cancer. And so if we added those two on together, you know, prostate and breast cancer are alarming. Okay? And virtually nothing is being done. Lots of research towards treatments and drugs, but nothing towards prevention. When did you hear anything about how to avoid things you might do to avoid breast cancer or prostate cancer, which really are caused by the same thing, because they're both estrogen receptors and are probably very sensitive to the same pseudoestrogens, xenobiotics, which involve a lot of the big business, like the plastics industry, the pesticide industry, the agrochemicals. Yeah.
Well, we've got to chop our breasts off now, haven't we?
You haven't got to, but it might be a good idea. So, lung cancer there, you see for males and females, even combined together, is still well less than breast cancer for just females. And then it drops down, you've got your bowel cancer here, which again is a substantial number, but only as much as male and females to the prostate cancer. And then practically everything else is, you know, really very small. You know, there's obviously a lot, but it's very small. So these are the main ones, the breast and the prostate, which would be undoubtedly have common links because they're just synonymous, and lung cancer and bowel cancer. Now, the bowel cancer one was interesting because the guy at the first milling company that I went to, he said, we don't do spelt here in bags, we do it as flour for you. But he said that you can go to Chappenham, I think it is, farm, which is not far from where we were, where we are in Devizes. And so I thought about that, and then I left the note on the kitchen table, and I thought, oh, I can't be bothered to go down into Somerset at this stage. And then I read an article from one of the colour supplements about it. And the chap who started it started Monsoon. You've probably all heard of Monsoon. And he made a lot of money, and he bought this farm down in Somerset. And his sister tragically got bowel cancer, and he looked into it, and there's definitely evidence that bowel cancer was linked to lack of fibre, good fibre in the diet, and that we'd lost the good fibre in the diet with modern wheat. So he put the whole field, the whole farm over to spelt. You could look it up on the internet, and they will send you packets. I've just got the first one, which I made that spelt with, 20 kilogram bags, which is not too big, and to make spelt. And so that's where that came from. Sharpenham Park. Sharpenham. I think that's it. I'll look it up to make sure. But what? Sharpham, is it? Yes. I'll just stick it on the internet or spelt or something, it'll come up with that. But his story was very interesting why he did this. And it's a lovely company. And that's all they do. And you ring up and you say, I'd like to have a 20 kilogram bag. And they say, when would you like it? And, you know, It's not expensive, you know.
It's grain or it's flour.
It's grain.
Yeah.
Yes, they'll sell you flour as well,
Yeah.
but I will get the grain if you can.
Doesn't it go rancid after 4 weeks?
No, it's the grain.
Oh, it's the grain.
It's the grain, yes, you can buy the whole grain. You see, the other ones wouldn't give me, they'll give me ordinary wheat grain, but this one was the only one I could find which would do you spelt in reasonable sacks, okay? Ever. Forever, because remember, the brand seals the grain so the germ inside doesn't go rancid. Okay, so you will never need to worry. You could go into a pyramid and be there for 5,000 years and the grain will come out and grow. You might not, but they would. Okay, so it was just interesting because I wanted to know, you know, where cancers were in relationship to diseases and also which were the most common ones. Oh, that's going fine. And these are the hot spots. So the areas which are in the darker colour are the hot spots of incidence of cancer, also which is interesting. And the latest research goes back to 1997. So you can see all this is way out of date, isn't it? But the heavy industrial parts of the northeast, northwest Manchester, Liverpool, and also this part down in Hampshire is not good. And look at this one, that's around the Bournemouth area, isn't it? Not good in Bournemouth. Lots of people retire to Bournemouth, but it's a hot spot there. And London's not as bad. And then there's a little, I guess that's Plymouth down there. Not a very good spot either. I was quite pleased, you know, the area where I live now, Marlow, is pretty low. So the question is why, isn't it? Is it to do with what people eat? Is it to do with the environment? With pollution and toxins? Is it to do with the emotions and so on? And this gives you, this is a difficult one I'm afraid to read. But this was the one that surprised me, is top of the charts in all the countries was Denmark. And the question is, why is Denmark top of the charts as far as cancer, with the highest incidence of cancer, all of you? It's very low selenium, yeah. Very important that you eat that Kamut wheat then, which is high in selenium. Okay, we were not too bad. I think, just trying to see on here. I think that's us there. We were actually not too bad down the list as far as the cancers. I think that's UK here with the females, and males were a bit better. I'll try and get one to blow up a bit better. Right, so yeah, I'll play that little film. That's an interesting one off Dr. McCullough's site about the business behind cancer. The average cost to patients in America is $50,000 to be treated for cancer, which includes the drugs and the medication and things. 50,000. And the number of people with cancer in America pushes this into a multi-billion industry, which has no intention at all of putting any emphasis into trying to find the cure, the cause. They want to promote more and more drugs and other ways. Otherwise, they're putting all cells out of work and the big business.
And that doesn't include the funeral costs.
No, it's just unfortunately, you know, what people will spend, which may be successful, may not. So when we did the seminar, when I first sort of put the thing together, it was rather a daunting task because it's something I've always wanted to do. But on the other hand, I thought when I started getting all this information, it was mind-blowing. Where do I start? You know, how do I present what the people might need and causes and so on? So after the first time we did this, I said we build a little flow chart. And this you'll find very useful because this is something you can do with your patients, and this is what I do with my patients. I check them around methodically for everything that I can. And patients like this, they come and see me because I do that. And I say, well, we'll check the hormones here, we'll check the toxins here and so on, and we work around it. And I'm not in any way a cancer expert. All I do is treat people who've usually had it or undergoing treatment at the present time, and do what you can. Okay, there's none of the here are going to proclaim as cures, but they're all things that can help people and give them advice, because they don't get any advice otherwise, and this is the sad thing. So we'll start off with a cancer cell in the middle. And I don't want to go into details what a cancer cell is. You're all familiar that it's one which has got uncontrolled growth and exposure of oncogenes or cancer genes. So all the emphasis in more recent years, since the discovery of DNA and RNA back in the 1950s, has been on mutations of the gene. So there's abnormalities in the gene which sparks off an uncontrolled growth, and the cancer cell seems to be basically a stem cell. So it lives forever. It's an eternal cell. And so it's a way the body promotes new tissue into that area. Unfortunately, the new tissue goes on growing, and once it bursts through its sort of encapsulated area, it may cause obstruction onto arteries and blood vessels, and then the people pick up infections, and that's what they die from. People don't technically die from the cancer, they die as a result of the pressures from the tumour. But if they can switch off that growth mechanism, then this is one way that they can be handled. So gene mutation is something that's looked at. And of course there's the inherited gene mutation and there's the acquired. So we may inherit our gene from our parents, and these are called oncogenes, or we may have acquired it through defects, particularly in methylation. Methylation, as we'll see, puts the caps on the genes and keeps bad genes from expressing themselves. So when we get methylation defects and deficiencies in methylation, we expose genes which we would rather not have done. Okay? So SAM is the universal methyl donor. SAM also helps to methylate hormones, particularly the estrogens, for breaking them down or metabolism. We know that we can get gene mutations from deficiencies in folic acid. You know, this happens with spina bifida and other diseases, but folic acid is really very, very important to stop genetic mutation. Zinc and all the other coenzymes fall into pattern to try and stop genetic mutations. On the other hand, for the last 80 years, it's been known that cancer cells are hypoxic. In other words, they suffer from lack of oxygen, and they tend to respire anaerobically as opposed to aerobically. So in other words, they work in that first part of making energy called glycolysis rather than Krebs cycle and electron transfer. So they work by glycolysis, so they're anaerobic. And as a result of the not being able to go beyond pyruvic acid, they shunt the pyruvic acid into making D-lactic acid instead of making acetyl-CoA in the mitochondria. So they are really very inefficient cells. They're immature and they're inefficient. And they have very low oxygen carrying capacity, and this is why we call it hypoxia. And this has been known for 80 years now, and is now gaining back in popularity as something patients can do, is to improve their hypoxia, and sometimes involved with even going to oxygen diving bells and things like they do for multiple sclerosis, hyperbaric chambers, but to increase the oxygen, increase the carrying capacity, along with a diet that decreases the glycolysis. So the more modern research seems to indicate that the ordinary cells in the body, the non-cancerous, can convert fats and protein straight into the Krebs cycle and use that for the production of energy, whereas tumour cells can't. Tumour cells rely upon glucose and only glucose. So starving the person of glucose, it was putting them on a glucose-free diet, which again has been known for something like 80 years. Daniel Quigley, who was a big medical surgeon in Omaha in America, talked about this back in the 1930s and wrote books on it. The only way you can effectively treat people with cancer, apart from the surgery, is you have to get them off sugar completely. So, and it's refined sugar. It's not raw cane sugar is okay, but it's the refined, particularly the fructose from high fructose syrup. So hypoxia is really, really important way of looking at this and putting people on the glycolytic pathway. Okay, now we know that the environment has a big important part. So chemicals, toxic metals, radiations, and certain infections like viruses can infect the gene. Okay? And they can affect very much the hypoxia because they can inhibit the enzymes to help us make, to get to the Krebs cycle. And of course they can create free radical reactions which will then lead towards damage to the gene. So that's why toxins and chemicals can have a big effect. Now hypoxia is also caused by deficiency. Now the most common things, you remember we did this with Sally when she showed up to eyes up and down, And the first thing I did was iron. Okay, iron checks for the hemoglobin. B12 checks for the size of the red blood cells. But as we gradually get into hypoxia, we'll learn there's lots of other things that can affect hypoxia. The essential fatty acids, getting the oxygen across the cell membranes. Making those cell membranes more permeable requires vitamin P, or what used to be called vitamin P, or bioflavonoids, okay, rutin, etc., and hesperidin, bioflavonoids, really part of the vitamin C complex, are very important. Vitamin B6 is very important because this helps to make hemoglobin in part of the heme molecule. So there's lots of things we need to look at with hypoxia. We know that the body should normally recognize a cancer cell and should destroy it. So we need to look at the immune system. So the immune response is things which will boost this person's immune system up. And one of the most powerful ones of this is vitamin C. And this has been known, high doses of vitamin C can increase the immune response. And you might want to use a straight C, a C which you can recycle with alpha-linolenic acid, that's the ALA, or maybe the C complex. Again, muscle test, which one is it? Silver, equal mix silver and B1, or thiamine, was very good. The immune response depends upon how active, how much energy your neutrophils and your T cells and white cells has got. And basically, if you are tired, which people very often are who've got malignancies, then the chances are your immune system is tired as well. So it's the same cell. If you're going to be deficient and tired in your muscles, you're probably tired in your white cells. So by looking at what boosts that energy up, and the main block, remember, is between pyruvate and acetyl-CoA, and that needs B1. So these tired cells often need B1, and if you give them B1, they come into life and they start spinning the Krebs cycle. Okay, within the, then they can produce the free radicals to kill the tumours and the infections. We know that the immune response can build up with melatonin or getting the pineal to work. The pineal has a profound effect on immunological responses. And finally here we know that cells, cancer cells, can self-destruct, called cell apoptosis. And this can be stimulated by vitamin D. So here we bring in vitamin D, whether it's from sunshine or taking it. Zinc, very, very important as part of the whole enzyme process of stimulating apoptosis. Coenzyme Q10. And coenzyme Q10, again, very, very important with the energy production in the body, the final part of making energy. But you'll find that CoQ10 really shows up well when you mix it with the sesame and the peanut oil. So the good CoQ10, which so many people need, is the oil-based one. Most of the commercially produced CoQ10 in oil base is in rice bran oil. And the trouble with rice bran is it's loaded with arsenic. Okay? And arsenic is one of the biggest poisons to the lactate pyruvate dehydrogenase enzyme to convert pyruvate into acetyl-CoA, just where the B1 works. So if you fill someone up with arsenic, it'll make their situation even worse. They won't be able to work aerobically, they'll be working anaerobically. You cannot, I haven't identified organic rice bran oil. You can get rice bran oil, no problems, but we were warned off rice bran with our colon cleanse unless you can get it certified organic, which we did. So we pay a lot more for that because everybody said if you use rice bran, which you can buy commercially, it's well known it's full of arsenic. Okay? Mainly because they use arsenic pesticides in the paddy fields. Okay? So you have to have certified organic. We know that melatonin has a profound effect on cell apoptosis or a lack of melatonin by people who particularly work with airplanes and are going through changes in time zones, and it messes up the pineal gland. So restoring the pineal body clock function by stimulating the melatonin stimulates cell apoptosis, by decreasing the toxins, obviously within their environment, and interestingly, tocotrienols, that second part to the vitamin E complex, are much more involved with apoptosis rather than the tocopherols. And one which is gaining interest all over the world now is black seed oil, otherwise known as black cumin oil, black cumin seed oil, which is what we'll have a look at the sheet of that probably later on or tomorrow. Okay? So that's a really interesting new oil that's coming in to help people for a host of things. but particularly for cell apoptosis. So that's the picture. So what I did is I just photocopy that and keep it in the treatment room, and when someone comes in I say, right, we're going to go through this, okay? So here's your problem in the middle, or that's where your problem was before you had it cut out. Now let's have a look on this side. We talk about genes. So let's have a look at a few deficiencies, whether there's a gene mutation. So how do we tell if there's a gene mutation? Do you remember? The SNP. Yeah, this is basic. We put on the 5 base substances, the RNA, DNA markers that are in your kits. You find the one which weakens, and then you find the one that negates, and there's your SNP. So your combination, you've got G going into T or A into and so on. So then you look up on your chart and say, that is a combination for methylene tetrahydrofolate or whatever it might be. Okay? So that's when you know you've got to give that coenzyme. So somebody who's got a gene mutation, this may be inherited. Okay? So it could be something, and this was the tragedy with Angelina Jolie, where you said where you have to have your breast cut off, is because she carried an hereditary disease, an inherited oncogene, BRCA1 or BRCA2, which meant she was highly likely because it was in her family. Okay? Now, sad that it might be, whether she made the right decision or not is not for us to, but she obviously went through that very, very difficult decision. And of course, my feeling is that when people have surgery and chemotherapy for the cancer, it hasn't actually addressed anything to do with the cause. It's chopped the result out, isn't it? So the sad thing is, in a lot of cases, it comes back, and it comes back somewhere else. Because if it's estrogen dependent, it will just come back on other tissues which are estrogen dependent. And that includes the intestines and the lungs and all sorts of estrogen receptors on many other tissues now. So getting help post-surgery, post-chemotherapy is very important, because this is what's missing with a lot of people, both males and females, for various cancers. They don't have the follow-up to say, well, this is what you could be doing, you know, and the fact that somebody will do it and help them is of great interest to them. So if they show positive to a SNP, then consider giving the coenzyme, and maybe if it's an acquired SNP, in other words, through some form of toxin, radiation, chemicals, virus, etc., we may need to improve that. And we know that folic acid deficiency leads towards changes in the cell structure of the DNA. So folic acid plus zinc, remember, to make all those different ones, or the other coenzymes. So most coenzymes are activated vitamin Bs, which you're familiar with, except the other ones are vitamin C is a coenzyme, SAM is a coenzyme, CoQ10 is a coenzyme, alpha lipoic acid is a coenzyme. So those 4 are not vitamin Bs. Okay. So we could have done a quiz, couldn't we, Jill, on that, of which coenzymes are not vitamin Ds. So when we were teaching last month in Hawaii for the Australian group, I said that we were talking about molybdenum, and I said that anybody knows the 3 molybdenum-dependent enzymes can get a free arthritis kit. 2 people got it right. Can you believe that? Okay, so straight out, boom, 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 aldehyde dehydroxylase, xanthine oxidase, so I thought, oh no. So right, so that was 2 kits. Okay, gene mutation. So that's the gene. So this can be acquired or inherited. And now we also know that we have protector genes in our system, suppressor genes they're called in our coding, and oncogenes. Now what you want to make sure is you keep the top on those oncogenes from expressing. Do you remember when I showed this many times, that what a gene is like is it's mechanically turned on by the receptor site of the agent that bonds onto the receptor site, or the ligand. Okay? And this literally changes the conformation by its polarity on the receptor site, and literally changes the shape of the proteins, and will pull into the gene, from the nuclear membrane there, and pull the sleeve. Now genes are made of proteins and DNA. Okay? Now the DNA is covered by the sleeve, which is protein. And when the sleeve is moved through the binding on the receptor site, it exposes a part of the gene. And this was the whole thing about the Human Genome Program, was it was expected to find that there were a certain number of proteins, one protein for every gene. There were 25,000 genes. So they expected to find 25,000 proteins. What they found is hundreds more, okay, or 100 and something thousand proteins, which means that each gene potentially can encode for a number of proteins. And the only reasonable explanation for this is it depends upon which part of the gene is exposed. So if we expose the hand, this has a message, because this is what the DNA is showing to encode for. If we show a bit more, it may encode for a totally different protein. And up here could be the oncogene exposed. So we can, depends upon which part of it is, and this exposing of the sleeve is mechanical, depending upon what the substance is binding to the receptor site. So if that's estrogen binding there, it will stimulate something to do with estrogen function. On the other hand, if it's a pseudoestrogen and it locks onto the receptor site and continues to expose that, then we continue to expose it, and it could be the oncogene. Okay, so you see the importance of why synthetic substances need to be, particularly synthetic hormones, kept to a minimum. So it's all to do with the exposure of that. Now, keeping the top on the gene and not letting that exposed is a methyl group, a CH3. And methyl groups are really very, very important from a point of view of stopping methylation defects. And many cancer experts say that cancer is a methylation defect. And my experience with this, as far as treating hormone-dependent cancers, is yes, it is. It's my finding is that they all strengthen to SAM. Okay, now SAM, as we'll find, is a substance made from methionine. So we'll come on to it and look at it in a little bit later. Methionine is an essential amino acid which goes on then to make S-adenosyl methionine, and to add the adenosyl part, we get that from ATP, adenosyl triphosphate. Okay? And the transfer enzyme there seems to be magnesium dependent, has everything to do with ATP is. But to make SAM, you don't give methionine, because if you give methionine, you add to the substrate. If they strengthen to SAM, it means they're not got enough SAM, which would mean relatively they'd have high methionine. So it doesn't make sense to give them more methionine, okay, because it would load it up, and that would build up without being able to convert. So we can give them ATP, but that could be used in 101 places in the body, or 1,001. What you need to do is to have the transferase enzyme. And there's one little plant that's got the transferase enzyme. Okay? And that's the magic, is finding what that plant is, which supplies that. So the plant is rosemary. So you will find everybody who shows positive to SAM, okay, will strengthen to rosemary, because that's what they need. It contains the enzyme. But you mustn't use the tincture, because the tincture is alcohol, and the alcohol destroys the enzyme. Okay? So it has to be the powder. Okay? Now, rosemary is a particular difficult herb to eat, because it's like a needle. So it has to be ground into a powder, fresh and encapsulated. Okay? The tincture doesn't.
Not dried. Must be fresh, not dried.
Oh, yeah, no, it can be dried.
Yes.
Yes, it can be dried, but it has to be not as a powder, which is old, in other words, because a lot of the valuable things in herbs are the volatile oils, and so the fresher you can get it, the better.
Yeah.
So the best is actually, I think, is to use the needles and grind it.
Yeah.
That's what I do. But it's a wonderful herb. It smells really nice, and you can sprinkle it on the food. You can swallow it as capsules. But anybody with SAM, and this is the secret, is rosemary is very, very good on these. Okay, it shows positive to them. Don't give methionine. SAMe has been shown not to be very stable, and many companies, certainly in the States, gave up using it because it's not viable after a month. So you buy this bottle, costs you a lot of money, bring it home, and a month's time it doesn't, even muscle test doesn't work. It's got no, it's not positive. So this is the trouble with coenzymes. They have a shelf life, and SAMe is particularly a short shelf life. Okay, so that's SAM. Now we also need SAM to methylate, because it's the universal methylator of our hormones, particularly the estrogens. So we know with the estrogens we need to hydroxylate, and then we methylate. And the biggest problem with the estrogens is whether they hydroxylate but we don't methylate. Okay, in other words, you get a build-up of hydroxylated derivatives, and that certain of those, particularly the 4 and the 16 of the estrone, are bad. They're pro-malignant ones. So we need to. So everything to do with breaking down the estrogens, the bad estrogens, everything to do with keeping the caps on the oncogenes is keeping the SAMI up. In other words, keep the SAM up. So we need to make sure that S-adenosyl methionine or methylation is looked after. So I find that shows up always with hormonal dependent cancers. And that's the big ones that we're talking about, the prostate and the breast. Okay? Yes, search for hereditary SNPs, supply the coenzyme that's necessary, but 9 times out of 10 the coenzyme itself is SAM. So it brings you back to that. Chemicals, toxic metals, yes, you can do that with the usual vials in the kits, the chemicals, toxic metals, radiation, infections. So let's just whip through this. Sorry? Phase 2 detoxification. Yes. Yeah, the major pathway for metabolizing estrogens, okay, and we're just talking about these types of hormones, is hydroxylation first, methylation, and then conjugation. Yes, there's 3 pathways. With hormones like estrogens and with adrenaline, noradrenaline, the catecholamines all go through 3 pathways. Okay, a xenobiotic, like a drug or whatever, will go through 2. They'll have a cytochrome P450 phase 1, and then could be methylation, could be sulfation, acetylation, and so on. But with hormones, and the ones that we're after are mainly the estrogens, the bad ones, we're looking at hydroxylated derivatives. So the hormone is hydroxylated, and then it can't methylate. So you get the build-up, and if it's a bad one. Now the trouble is that certain genetically, people have a bigger predisposition to the bad one, which is the 4. So the 4-hydroxy as opposed to the 2 or the 16. The bad one is the 4. The 2 is the good one in estradiol and estrone. But in estrone, the 4 is bad and the 16 is bad. Okay? But in estradiol, the 4 is bad, but the 16 is good. It's known as estriol. Okay, so the 4 is the really bad one. And unfortunately, the people who've got the greater propensity to do it are the blue body types, because they've got the short straw as far as their enzyme expression of that particular one. So here what we found is they all weaken to I am loved. So from the emotional point of view, with all cancer cases, they don't feel loved. And this may be the root cause of what triggers the response. And we know that when people do weaken to that, and we measure the hypothalamic hormones, they have low oxytocin, because oxytocin is that hormone associated with love and bonding, isn't it, between mother and child, and between people. It's the love hormone. And they get high corticotrophic releasing hormone. So the hypothalamus does this. So the oxytocin goes down to the feeling. So people are actually changing their chemistry in how they feel. And these people come in possibly into the world feeling like this to begin with. This lowers the immune response and lowers the natural killer and T lymphocytes. So you're off to a bad start. So the common features I find with hormonal ones, leaving aside the mind, are methylation defects and immune system responses. Okay, they're not activating their immune system enough, and they've got methylation defects. So here's a chart. This is on our website, and you can download it. It's just a color chart of the cytochrome P450 hydroxylation pathways, and it's color-coded into red, green, and blue, of which are the ones which are defective. Okay, so you can cross-check and say, I'm a green, and you'll see that something like 75% or more, 80% of drugs go through these pathways. So there is charts, and if you're interested, you can just download this, of drugs that we know we've got the short straw. So I'm a green. We're very sensitive to probably 70-80% of chemicals in the environment, whereas blues are chemically sensitive, particularly to pesticides, herbicides, hormonal-like ones. Reds are the best of all of these.